In this video tutorial, I'm going to discuss about isoenzymes, a very important concept in biochemistry that explains how one reaction can be carried out by multiple versions of an enzyme. So what exactly are isoenzymes? In simple words, isoenzymes are closely related enzymes that catalyze the same chemical reaction, but they differ in their amino acid sequence and structure. That structural difference makes them behave slightly differently inside our body. Here is an easy way to visualize it. Think of isoenzymes like different vehicles. A car and a bus both take you from point A to point B. Same function, right? But they have different engines, designs, and performance. Similarly, isoenzymes catalyze the same reaction, but their internal design, that is, their molecular structure, differs. And because of that, their behavior or properties also differs. Now, since their molecular structures are different, isoenzymes also show different physical and chemical properties. Let's quickly enlist these major different properties. First, because of their structural difference, isoenzymes carry different overall ionic charges. And when we use a technique like electrophoresis, where molecules move through a gel under an electrical current, this charge difference makes isoenzymes migrate differently. Second, isoenzymes have different tissue distribution. That means different tissues or organs preferentially express a specific isoenzymes depending on their metabolic needs. Third, they differ in heat stability. Some isoenzymes can tolerate higher temperatures while others lose their structure and activity when heated. Fourth, they have a different sensitivity to inhibitors and activators. One form of enzyme might get inhibited by a particular substance, while another form of the same enzyme keeps working perfectly fine. Fifth, isoenzymes differ in their Km and Vmax values. In other words, they don't bind their substrates with the same affinity and they don't work at the same maximum speed. This means that in different tissues, some isoenzymes are more active under certain conditions than others. Sixth, some isoenzymes even differ in the cofactor they need to function optimally. And finally, isoenzymes are immunologically distinct. That means our immune system recognizes each isoenzyme form as a slightly different antigen. This property is especially useful when we use antibodies to identify specific isoenzymes in diagnostic tests. So all these differences, charge, stability, kinetics, and more arise because isoenzymes have different structures. The only thing that they share is their function. They catalyze the same reaction, everything else can be different. Now that we understand what isoenzymes are and how they differ in properties, let's move one step further and see how do these isoenzymes actually form. Because their structural difference must come from somewhere, right? So formation of isoenzymes. There are three main mechanisms by which isoenzymes are formed, and each mechanism explains a different reason why multiple versions of an enzyme exist. The first mechanism involves different genes, distinct genes producing enzymes that catalyze the same reaction. Isoenzymes formed this way are called true isoenzymes. To understand this better, we will look at two scenarios, one for monomeric enzymes and one for oligomeric enzymes. Let's first take monomeric enzymes. These have a single subunit, meaning just one polypeptide chain. Here, different genes code for a different single polypeptide chain enzymes. For example, suppose enzyme 1 and enzyme 2 both catalyze the same reaction, but they have slightly different amino acid sequence as both code by different genes. Let's say gene 1 and gene 2. That means both are isoenzymes, and since they come from different genes, we call them as a true isoenzymes. Classical example includes the isoenzymes of aspartate transaminase or AST, one found in cytosol and the other in mitochondria, both coded by different genes. Similarly, malate dehydrogenase or MDH. It has a cytosolic and mitochondrial form. Again, distinct gene, same reaction. And another good example is amylase. Salivary amylase and pancreatic amylase both break down starch but are coded by different genes in different tissues. Now let's look at the oligomeric enzymes. These are enzymes made of more than one subunit. Each subunit is a separate polypeptide and each can be coded by different gene. Let's understand this with a very important enzyme, creatine kinase or CK. It exists as a dimer, meaning it's made up of two subunits. Each subunit can be either an M type, which is coded by gene on chromosome number 19, or a B type, coded by a gene on chromosome number 14. Since creatine kinase is a dimer and we have two options for subunit, three possible combination can exist: MM, MB, and BB. This gives rise to three isoenzymes. Creatine kinase MM, found mainly in skeletal muscle. Creatine kinase MB, found mainly in cardiac muscles, and creatine kinase BB, which is found mainly in the brain. And because each subunit comes from a different gene, these are perfect examples of true isoenzymes. Another classical example is the lactate dehydrogenase or LDH. It's a tetramer made up of four subunits. Each subunit can be either H type, which is coded by gene on chromosome number 12, or M type, coded by gene on chromosome number 11. Remember, this M subunit in lactate dehydrogenase has nothing to do with the M subunit in creatine kinase. They are completely unrelated. They just share the same letter for naming convenience. This must be clear. Don't ever get confused on this aspect. Since lactate dehydrogenase has four subunits and each can be H or M, there are five possible combinations of this subunit, forming five different lactate dehydrogenase isoenzymes. These are: all four subunits can be H subunit. This makes LDH1, found mainly in heart and RBCs. Then combination of 3H and 1M makes LDH2, also found in heart and RBCs. Combination of 2H and 2M makes LDH3, found in lungs. Combination of 1H and 3M makes LDH4, which is found in kidneys and placenta. And combination of all 4M makes LDH5, found in liver and skeletal muscle. So there are 5 LDH isoenzymes, each with different subunit composition and tissue distribution. Again, since their subunits are coded by different genes, LDH isoenzymes are also true isoenzymes. We will have a detailed discussion about these isoenzymes in next subsequent videos. Here I am just superficially discussing these enzymes. Okay, so that was the first mechanism, true isoenzymes formed by different genes. Now let's move to the second mechanism, which involves variation within the same gene. This involves a different allelic form of gene in a different individuals. The isoenzymes created by this mechanism are called as allozymes. Remember, if different genes participate, we call them true isoenzymes. And if different allelic forms of genes participate, we call them as allozymes. Now I know you must be wondering, what does this different allelic forms actually means? See, every gene sits at a fixed position or a fixed spot on a chromosome. That spot is called its locus. But here is the interesting part. The same gene can exist in slightly different versions from one person to another, and those versions are what we call alleles. So when we say different allelic forms, we are talking about the same gene sitting at the same locus but having small variations from one individual to another. For example, think about the gene for eye color. It's located at the same place or locus in everyone, but one person may have an allele for brown eyes, another for blue, or someone else for green. So I think concept of allele is clear to you. So now basically, in this mechanism, the enzyme comes from the same genetic locus, but because of different alleles, they have a small structural difference in different individuals. They still do the same job, but their structure aren't exactly identical. So in other words, allozymes are variants or variations of the same enzyme found in different individuals across a population. One real-life clinically important example is glucose 6-phosphate dehydrogenase or G6PD. Researchers have identified about more than 400 different allelic variants of this enzyme across the human population. Now each individual person has only one form, but when you look at the entire population, you see these all forms. Now let's look at the third and final mechanism of isoenzyme formation. This involves a post-translational modification And isoenzymes formed by this mechanism are called as isoforms. Think of this like customizing a car after it leaves a factory. The base model is the same, but you can add different accessories, right? A seat cover, a floor mat, phone holders. And each customization creates a different version of the same car. Similarly, the same enzyme polypeptide gets modified after it's made, and different modifications create a different isoforms. One very good example is alkaline phosphatase or ALP. Once alkaline phosphatase is synthesized by a process of translation, different numbers of sialic acid residues attach to them, creating a different isoforms of this enzyme. So once again, they have a different structure but same function, right? Now that we know how isoenzymes form, the next question is, how do we actually separate and identify them in the laboratory? There are four main techniques, and each one exploits a different properties of isoenzymes. Technique number one is electrophoresis. Remember how isoenzymes have a different structure? Well, different structure means different ionic charges. Now in electrophoresis, proteins with different charges move at a different speed through a gel when electric current is applied. This separates them into distinct bands. For example, when we run lactate dehydrogenase on electrophoresis, it separates into 5 distinct bands, each representing one of the 5 lactate dehydrogenase isoenzymes we talked about earlier. Technique number 2 uses antibodies. Since isoenzymes are immunologically distinct, they can be recognized and precipitated by specific antibodies. This is actually how we routinely identify creatine kinase MB isoenzyme in clinical labs. We use antibodies specific to the M subunit to detect creatine kinase MB isoenzyme. Technique number 3 involves chemical inhibitors. Different isoenzymes have different sensitivities to various chemical inhibitors. A classical example is acid phosphatase or SCP. The bone isoenzyme form keeps working even when you add tartrate, means it is not inhibited by tartrate. But the prostate isoenzyme form gets completely inhibited by tartrate. This property helps us determine the source tissue of the enzyme. And the fourth technique is heat inactivation. Some isoenzymes are heat stable while others denature at high temperatures. They have a different heat stability. Here is a clinical useful example. Alkaline phosphatase alpha 2 isoenzyme of placental origin remains stable even at 65 degrees Celsius, but the isoenzyme of hepatic origin loses all its activity at this temperature within 30 minutes. So if we heat a sample and the activity remains, we know it's the placental form. So these are main 4 separation, or you can say identification technique for isoenzyme. So let's rapidly recap the key points covered in this video. Isoenzymes are enzyme variants that catalyze the same reaction but have a different structure and properties. They form through 3 mechanisms: distinct gene creating true isoenzymes, allelic variations creating allozymes, and post-translational modifications creating isoforms. We can separate them using electrophoresis, antibodies, chemical inhibitors, or heat inactivation. In next video, I will discuss about common isoenzymes and their clinical usefulness in diagnosing diseases. So that's all for this video. I hope that all the concepts are clear. If you have any query or confusion, please write it down in the comment section below. Thank you. See you in the next video.